Good day, everyone. Thank you for attending today's webinar on anti-progestin therapy targets hallmarks of breast cancer risk. We're very excited to bring this webinar as part of our series on VitroGel for ex vivo models. I am Christopher Oak from the Well of Bioscience, and I will be your moderator alongside our speaker for today, Dr. Alejandra Ferrer-Diaz from the Well of Bioscience, and our guest speaker, Dr. Bruno Simões from the University of Manchester, UK. A few items before starting. During the webinar, you can utilize the Q&A and any questions you have. We will collect them and present them during our Q&A session after the talk. We'll have some quick poll as well during the segments. During the webinar, feel free to participate as we go along. Also, the webinar will be recorded and can be accessed later for your convenience. Excited to start this. So first up, we're speaking is Dr. Alejandra Ferrer-Diaz from the Well of Bioscience. We'll start with a short introduction on VitroGel and the applicational uses. Afterwards, we will turn it over to Dr. Bruno Simões to present his research and the university research on breast cancer models. Ali?
Thank you so much, Christopher, for that, and thank you everyone for joining us today. So today we'll talk about the role of VitroGel hydrogels for ex vivo patient-derived organoids and tissue explants. VitroGel hydrogels are a xeno-free synthetic system that provides an optimal xeno-free environment for ex vivo culture applications. When using VitroGel, you can be confident that you can preserve the phenotypical properties of ex vivo microstructures in organoids. Because VitroGel is a synthetic system, it enables reliable disease modeling, personalized medicine, and drug screening. And again, because it is synthetic, one of the things that you can expect when using a system like this is that you will achieve high reproducibility and consistent. In the second part of today's webinar, Dr. Simões will highlight how he leveraged the VitroGel system to establish breast tissue-derived organoid cultures for his study. At the Well Bioscience, one of our main focus is to develop 3D advanced models that can drive the advancement of biomedicine. And we focus on 3 main pillars. One of them is precision medicine, cell therapy, and then manufacturing. For precision medicine, the idea is to establish fully integrated xeno-free workflows that can facilitate the advancements of 3D models. And as a result, that could lead to the development of drug discovery platforms and personalized medicine. In terms of cell therapy applications, the idea is to develop organoid models in xeno-free cell preservation and utilize our platform as an injectable carrier for therapeutic delivery in clinical applications. And for the manufacturing part, the notion is to develop next-generation scalable xeno-free platforms with our hydrogels to then that this could be used for biomedical use. One of our main ideas, and we always tell people this, is that it's very important to start with the end in mind. And that means selecting the right platform and the right materials for your research that can then be easily translatable for clinical applications. And our mission as a company is to unlock the full potential of 3D cell technology to then enable clinical translation and laboratory automation. At Well Bioscience, we not only provide hydrogel systems, we also provide an end-to-end platform for 3D cell and organoid cultures. And we provide the full workflow for downstream analysis and laboratory automation capabilities. And as I mentioned, one of the things that we focus heavily is on the production of VitroGel xeno-free hydrogels. And we have 2 different types of hydrogels. We have the ready to use, and I'll elaborate a little bit on that a little bit later. But essentially, the idea for this is that this is a very easy to use system, that the only thing that you have to do as an end user is add medium with cells or organoids and combine it with the hydrogel. And the ionic molecules will facilitate the formation of the gel. We also have tunable high concentration hydrogels, and this is what we call the building blocks. It allows you to, as a user, to have a little bit more control over the matrix by manipulating and adjusting the mechanical strength. In addition to that, we have the functional assay kits, and we have many of these, but I just talk a little bit about a few. One of them that we have is the VitroGel epithelial to mesenchymal GBM kit, and we also have the angiogenesis assay. So the idea is to provide you with this system that you can do from beginning to end. Besides that, we have the rocket cell xeno-free media. So the notion is to provide you with the full platform and xeno-free system medias that you can use for your cell culture applications. For that, we have multiple kits listed here, and these are what we call an all-in-one complete medium kit. Once you have this kit, you can establish your model, and you can do it without having to think about what else to add, because everything is all in one. You have everything you need to establish that. And we have these for iPSCs, for mesenchymal stem cells, for neural stem cells, organoid medium, and intestinal organoid kits. Besides the hydrogels, we also offer vitro inks for those of you who are interested in the 3D bioprinting realm. Additionally, we also offer premium grade quality growth factors that are a research-friendly price, and these could be used in combination with our hydrogel system so you can elevate your 3D-based cultures. Furthermore, we offer 3D cell culture burst vessels under our VitroPrime brand. And right now, we recently launched the VitroPrime 3D culture and imaging plate, which features a glass bottom and a medium exchange channel for easier 3D cell culture applications. We also have the spread attached plate, cell culture inserts, which are very important for invasion assays, and ultra-low U-shape attachment plates, and these are very important for spheroid formation and other types of models. Additionally, we offer self-recovery solutions. And think about this: when you have established your 3D cultures, one of the important things is to be able to retrieve those cells from the matrix and then perform downstream analysis. And that's why we offer the VitroGel organoid recovery solution and the VitroGel self-recovery solution. For downstream analysis, we offer the Cyto 3D LiveFed assay kit, a dual LiveFed assay kit that allows you to measure in real time the amount of live cells and dead cells that you have within your culture system. And with that being said, as I mentioned, we offer a Xeno-free complete workflow for the advancement and the establishment of 3D cell models. And now I want to dive in some of the properties of our VitroGel system and why it is so unique over other types of matrices that are in the market. As I alluded to earlier, VitroGel hydrogels are Xeno-free, no animal-based components. They're characterized, they're biocompatible. But one of the main applications of the hydrogel is that you have full room temperature operation. When you receive the hydrogel is liquid at room temperature, and the protocol is 20 minutes. Gelation is not dependent on temperature, it is ionic-based. A great feature of our hydrogel system is that it's tunable. You can adjust the mechanical strength to see how mechanical strength affects cell growth. And Dr. Simões will talk a little bit about that and how he leveraged this property for his own study. VitroGel hydrogels are injectable. It can be used for xenograft models, but because of the fact that they're not temperature sensitive, it can be used for lab automation. They're imaging friendly, and over here, you have some images that we have taken of cells and organoids within our matrix. And you also have easy cell harvesting. With our VitroGel organoid recovery solution, you can take your cells out of the matrix, and this factor is also compatible with animal-based matrices. These are some of the applications that can be achieved with our hydrogel system. You have organoid establishments, you can do 3D neurons, tumoroids, stem cells, invasion assays, angiogenesis, and beyond. Our products have been used in more than 1,000 research labs worldwide. It has been tested in more than 500 cell types, and we have 330 plus publications that use our products for their research. Now I want to compare and dive in on how our VitroGel hydrogel is superior over animal-based CCM. And I already touched base in some of these properties, but I want to reiterate them in order for you to understand how powerful our system is. So let's start with composition. As I mentioned, our hydrogel is synthetic and is pure. Animal-based ECMs, on the other hand, are derived from ECM tumors. And as you know, tumors are very heterogeneous, so that means that you're adding a lot of variability in your system. These matrices have over 2,000 undefined compounds, and because of that, you cannot achieve batch-to-batch consistency. Versus with VitroGel hydrogels, because they're synthetic, you have full batch-to-batch consistency. The preparation time with our hydrogels is less than 20 minutes. With animal-based CMs, it's more than 2 hours. Our system is room temperature stable, so you have a room temperature protocol versus animal-based CM. ECMs, as you might know if you're a user, you know that they're temperature sensitive and you need to use an ice bucket. You have to chill your pipettes in order for that matrix to not polymerize. Our hydrogels are tunable versus animal-based ECMs are non-tunable. And because of the temperature stability part that I mentioned for our hydrogels ship at ambient temperature. And in addition to that, because they're synthetic, they're an ethical product. No animals-based ECMs are harmed. No animal are harmed during the process. And this aligns with the NAMS initiative that is established by the FDA and the NIH. When using our hydrogel matrix, you can save up to 80% of operation time and 40% of cost per test. I invite you all to scan the QR code listed here to explore the more than 20 advantages of VitroGel over animal-based extracellular matrices. Now I want to discuss a little bit about this line of product that we have, the VitroGel High Concentration Kit. And the reason for that is because in his study, Dr. Bruno Simões used the VitroGel Co, and he'll elaborate a little bit on that later. But I want to highlight how flexible this line of products is, and what we call the VitroGel High Concentration Kit as the building block of this hydrogel system, and it allows you to achieve unlimited creativity and maximum flexibility. And the reason for that is that with the VitroGel High Concentration Kits, you can tune the hydrogel strength. You can adjust it as shown here, and that can be achieved with the VitroGel dilution solution that is included within the kit. You have full control of the extracellular matrix composition. These hydrogels have various biofunctional groups, and the good part of that is that you can then understand how the functional group affects cell growth. A great property of the VitroGel High Concentration Kits is that you can do what we call the mix and match. And you can create and customize your own hydrogel within the lab. So let's say if you want to study how VitroGel RGD, the RGD peptide, affects growth, but you want to combine that with the collagen one, you can do it to then achieve and elevate your 3D cell culture application. And now I will highlight some examples of what you can do with the VitroGel High Concentration Kit for 3D cell culture and in 2D hydrogel coating. And I'll just go over this a little bit briefly for you to have an idea and understand the workflow. So for 3D cell culture, it's pretty simple. The first thing that you have to do is combine the hydrogel with the dilution solution to adjust mechanical strength. After you have done that and achieved your desired mechanical strength, then you can combine the hydrogel with your cell suspension. And as I mentioned, the media that you have for the cell suspension is what allows that gel to cross-link. Then after you have mixed properly, then you can add that to your well. You'll have your 3D cell culture established, and then you can just incubate for 10 to 15 minutes to achieve gelation. And after that, you add remaining media on top to ensure that there's proper nutrition, and your 3D cell cultures are established. So as you see, the 3D cell culture procedure is very easy, room temperature operation, and it only takes 15 to 20 minutes to achieve. Another great application is what we call the 2D hydrogel coating. And this is very interesting for people who do not want to transition to 3D right away, but do want to establish 2D cultures that are a little bit more elevated than your traditional 2D based cultures. And in this case, again, workflow is pretty simple. You adjust the mechanical strength of the hydrogel by combining with the dilution solution. Then after that, you combine the hydrogel with medium, no cells included. Remember, the medium is what facilitates the cross-linking, the gelation part. Then you add that into a well, let it incubate for 10 to 15 minutes at room temperature. And then after that, you just add more media on top with your cell suspension. And then the cultures are established, and the cells, as shown here, will grow on top of. Now I will provide an example of an elevated and of an advanced 3D model that can be achieved with our VitroGel high concentration system. And this was done by some scientists at the University of Manchester. And this is a tissue explant and vivo model, and I invite you all to scan here the QR code if you want to learn more. And in this case in particular, what they did is something that we call the sandwich method. And for that, you just use the VitroGel high concentration hydrogel, create a layer of that hydrogel. In this case, the researchers, what they did is that they took some tissue from the breast, embedded it in the hydrogel, and then add another layer. And they were able to culture this for over 7 days, and they demonstrated that the phenotype of the breast tissue was retained within our VitroGel hydrogel system. So now you can see that besides just culturing cells, you can create advanced models such as this ex vivo SPLEN model to elevate your research and make it more clinically relevant. Over here, I'm providing more examples of these ex vivo organoid and tissue SPLEN models that can be achieved with our hydrogel system. One of them over here highlights the use of VitroGel MSC, another type of hydrogel that we have for colorectal tissue. The next one is for VitroGel RGD for breast tissue. This is the one that I just explained. And this one highlights VitroGel RGD for lymphoid tissue culture, ex vivo cultures. So as you can see, our system has been validated across multiple tissue explants. And we have other customers who are also trying. This is just an example and a snapshot for you to see of what can be done with the VitroGel hydrogel system. With that being said, I will pass it along to Dr. Bruno Simões so he can explain to us his study, and now we can see how amazing is the study that he performed.
Thank you, Ali, for that VitroGel overview. Having explored how VitroGel can advance treated breast cancer studies, let's see how it's examined in a real-world application. Bruno, if you can share your slide there, let me do that. Yep. And today, really pleased to welcome Dr. Bruno Simões, a branch of cancer cell biologist with extensive expertise in breast cancer risk and prevention. Dr. Bruno earned his degree in microbiology and genetics from the University of Lisbon and completed his Ph.D. in Spain, where he studied how estrogen influences human breast stem cells. He later joined the University of Manchester, where his work focused on breast cancer stem cells and their role in resistance to endocrine therapy. He now leads laboratory research using normal breast tissue from prevention-focused clinical studies, aiming to uncover the earliest molecular and cellular events that drive tumor initiation. His research is dedicated to advancing chemoprevention strategies for women at increased risk of breast cancer. Dr. Bruno has authored over 30 peer-reviewed publications with more than 3,500 citations and serves as an associate editor for the breast cancer research. We're excited to have him share his insights with us today. Dr. Simões, we will turn it over to you.
Thank you. Can you hear me? Thank
Yes,
you. Can you hear me?
Yes, we do.
Thank you. Brilliant, Christopher. Thanks so much for the introduction, Christopher and Alejandra. And thanks for all the Well Bioscience team for the invitation. It's a pleasure to present our work, which is titled Anti-Progestin Therapy Targets Hallmarks of Breast Cancer Risk. And this work has been ongoing for 10 years, but recently culminated in a publication in Nature. And that's the work that I will show you today. But first, and before starting and going into our own work, I'd just like to remind all of us that despite all the advances in the screening and treatment, the number of women that die from breast cancer remains really high. And we can see here in this graph the absolute number of women that die per 100,000 women in the G7 countries and Australia. And as we can see, there are, for example, some countries like Italy and Japan that actually has shown an increase over the last 15 years in breast cancer mortality. But actually there is a good exception. The UK is one that shows a decrease in the number of women that have been dying from breast cancer. There is also this recent publication published last year that looked at the incidence and mortality of all cancers. And it has reported, as you can see here, that in the breast cancer in the adults aged 40 to 64 years old was the largest contributor to cancer incidence. And this is shown here, the breast cancer in the light purple. But it was also in the 2023 was also the second largest contributor to cancer mortality. The first contributor to cancer mortality is lung cancer, but then breast cancers is in this adult age group of 4 to 64 years old is the second largest contributor. When we just look at women, you can, this study also reports that breast cancer is the most common incident cancer in 164 countries, and this is also the most common cause of cancer death in 134 countries. So all this data, basically what is telling us is that, as Erasmus told us more than 500 years ago, that prevention is better than cure. So what we really need is effective breast cancer prevention strategies and drugs. And to do that, what we also need is to, first of all, to understand well how the development of the normal human breast can lead to breast cancer. And we know that the breast development occurs mainly in puberty and in adulthood, with the menstrual cycles and the effects of these hormones called estrogen and progesterone that lead then to the formation of these terminal ductal lobular units. So very broadly speaking, we can say that in the mammary gland there are two major epithelial cell types, the luminal cells and the myoepithelial cells. And I would like just to draw your attention to this selection of cells within the luminal cells that are called the luminal progenitor cells, because these are the cells of origin of most breast cancers. So, and why did we focus also on this study on the progesterone signaling? We focused on progesterone signaling because we know that there is an increase in the proliferation, the number of proliferative cells in the normal human breast during the luteal phase of the menstrual cycle, and this is the phase where the progesterone levels are higher. And we can see this here from this study done in Manchester in the 80s, in which we are seeing the number of proliferating cells in women aged 15 to 35 years old. You can see that the number of proliferating cells is higher during the luteal phase where progesterone levels are higher. We also know from my studies that when you have a knockout of the progesterone receptor, that it clearly shows that progesterone receptor is needed for the normal mammary gland development, in particular for the side branching and the alveolar development. So regarding its mechanism of action, we know that progesterone is a paracrine regulator of the luminal progenitor cells. And in the presence of progesterone, what happens is that progesterone receptor in the luminal mature cells is activated and then leads to the secretion of ligands like rank ligand that stimulate the proliferation of these luminal progenitor cells. And we know that, as I said, that these luminal mature cells are characterized by the expression of estrogen receptor and progesterone receptor, but these luminal progenitor cells are characterized by the expression of this marker, this transcription factor called SOX9, and if they are proliferating, they also are Ki67 positives. So we have questioned what was the expression of these markers, if it was different or not in women that would be at increased risk of breast cancer. And when we compared women that are at tissue from women that are at normal risk with tissue from women that are at increased risk of developing breast cancer, we found that there was increased expression of progesterone receptor, increased expression of this luminal progenitor marker SOX9, and also increased expression of the proliferating marker Ki67. Interestingly, we also found that there was an increase of the double positive cells for SOX9 and Ki67, which indicates that we have increased luminal progenitor cell proliferation. So in this diagram, I'm trying to depict this. So what we observe in women that are at increased risk, just we have an increased number of these cells here in green that have SOX9 and they are proliferating, so they are Ki67. And this is likely to be due to the signals, that the paracrine signals that they are receiving from the mature luminal cells that express the estrogen receptor and progesterone receptor. So we've questioned, can we target the luminal progenitor cells by targeting these paracrine signalings that are coming from these mature luminal cells? And we start looking first of all at epidemiological data. And we already know from many prevention clinical studies that anti-estrogens like tamoxifen, which is a well-known anti-estrogen drug, they can reduce the incidence of estrogen receptor positive tumors, but they cannot reduce the incidence of the most aggressive estrogen receptor negative breast tumors. And actually, if you see here, there is actually a marginal increase in the incidence of estrogen receptor negative tumors when these women receive these anti-estrogen drugs. And so this is likely the reason why anti-estrogens have been shown to breast cancer incidence, but not breast cancer mortality. We also know that progesterone, it does increase the risk of breast cancer more than estrogen alone. And we know this from epidemiological studies looking at women that have received hormone replacement therapy. And you can see here the data of women that have received these therapies for 5 years. And this effect, as you can see, unfortunately, is carried over for many years after these women have stopped receiving progesterone. Even after having stopped receiving progesterone for 5 to 8 years, they still are at increased risk of developing breast cancer. So we question at this time if the inhibition of the paracrine signals that were coming from the progesterone receptor positive cells could reduce the proliferation of these luminal progenitor cells and therefore reduce the risk of these estrogen receptor negative breast cancers better than the anti-estrogens. So to address this hypothesis and to address this question, what we have done was to set up a clinical study, which is called the Breast Cancer Anti-Progestin Prevention Study. And in this study, we have recruited 24 premenopausal women, all at increased risk of breast cancer. And we have performed MRI scans and then collected biopsies at the baseline and also 12 weeks after these women have received this anti-progestin drug called ulipristal acetate. They've received it 5 milligrams daily. So the first results we looked at was at the MRI scans, and we could confirm that the anti-progestin drugs were actually reducing the fibro-glandular volume. So this means that there was a reduction of the breast glandular density. And we know that mammographic density is a key indicator of breast cancer risk. But we did not know what was the mechanism. How is this reduction of this density occurring? So we then went on and analyzed the tissue that we collected using vacuum-assisted biopsies, which allow us to get enough tissue in order to look at different molecular changes that were occurring in response to this anti-progestin therapy. And we looked at different endpoints that I will show you today. We looked at epithelial proliferation, we looked at luminal progenitor fraction and activity, we also looked at single-cell RNA sequence and bulk RNA sequencing. We also did proteomics and image massing cytometry in the tissues, and then we also looked at collagen organization and stiffness on the tissues before and after treatment. So regarding proliferation, which was our primary endpoint, we found that there was a significant reduction of Ki67 after this anti-progestin treatment, and this is observed in almost all of the patients. So the percentage of SOX9 positive cells, which is a luminal progenitor marker, as I mentioned, was also reduced after treatment. And this was also the case when we looked for SOX9 and Ki67 double positive cells, which indicates a reduction of these luminal progenitor cells and the luminal progenitor proliferation. We then looked at the luminal progenitor cell population by flow cytometry. We used 2 markers that are CD40F EpCAM, which allow us to discriminate all the epithelial cells, the luminal progenitors, the luminal mature cells, and the basal cells. And we found that there was a significant reduction of the luminal progenitor cells after treatment, as we can see here in these representative FACS plots that I've put here, showing a reduction of the double positive cells for both EpCAM and CD40NF, representing the boxes in red. So we also then investigated the luminal progenitor cell activity and by performing a mammosphere colony formation assay. Basically, what we do is to dissociate the tissue, get the single cells, and plate cells at very low cell density in suspension, and then check for their formation of spherical colonies that we call mammospheres, which is a measure of luminal progenitor cell activity. And when we did that, we observed that there was a reduction of the capacity of these cells to form mammosphere colonies in suspension after the anti-progestin treatment. So the next question we wanted was to understand how was anti-progestin treatment really reducing the luminal progenitor numbers and their activity. And to address this question, what we wanted to do was to investigate the transcriptional changes in the different breast cell types after anti-progestin treatment. And we decided to do this by using single-cell RNA-seq analysis, which we were able to do for 6 paired samples, and we acquired data for more than 115,000 cells. And the first thing we did was to identify the 3 epithelial populations present, the luminal mature cells, here reported as luminal hormone sensing, the luminal progenitors, here reported as LASP, and the basal cells, myoepithelial cells, here reported as BMAIO. We also identified all the stromal cells, as you can see, different populations, the immune compartment, the fibroblasts, endothelial cells, and also the perivascular cells. And we identified all these cells using different markers, as you can see in the right, in collaboration with the group of Walid in Cambridge. And as you can see, I've highlighted the markers for the luminal progenitor cells. If you can please press the next. So the luminal progenitor cells showing that there is an increase of SOX9, that is the marker we have been using to identify these luminal progenitor cells, but also markers such as ILDH183 and KIT. So at this stage, we questioned ourselves which genes and pathways were differentially expressed in each cell population after the anti-progestin treatment. And we have looked at the differentially expressed genes on each of the populations, and we found that the majority of the genes that were differentially expressed were actually downregulated. And we could see that most of these genes that were downregulated were on the luminal mature cells, the LHS cells, the luminal hormone sensing. And this is, of course, not surprising, because these cells are the ones that express progesterone receptor, which is the target of the drug that we are using in the clinic, the ulipristal acetate. Next, please. So we also then looked at the proportion of the luminal progenitor and LASP cells within the epithelium, and we were able to confirm the reduction of the luminal progenitor cells even in our single-cell RNA-seq data, as you can see in here. Then we've decided to use a software called cell chat analysis to investigate the cell-cell communication interactions. And we identified that in the fibroblasts and the BMAIO cells, those were the ones that had the highest cell-cell communication interactions, both looking at the incoming interaction strength, the receiving signals, but also at outgoing interaction strength, the sending signals. So this just to highlight those cells. And then by using the cell chat analysis again to look at the interactions between these myoepithelial cells, BMAIO, and the fibroblasts, the different cell populations, we found that the collagen signaling, as you can see here highlighted in red, was the pathway that was most markedly downregulated after treatment, both as sender cells, outgoing strength, but also as receiver cells, incoming strength. So we then wanted to understand investigate which collagen genes were downregulated on each of these basal and fibroblast cell populations, and we also inferred the differential collagen ligand receptor interactions with the other cell types, and the ligands are here represented in the bottom, all the collagens, and the receptors at the top, we could observe that there was a reduction of both the autocrine signal to those same cell types, but also as a paracrine signal to the other cell types. And I would like to highlight, next please, that the collagen VI genes were found in all of these cell types as reduced genes after this anti-progestin treatment. So we also, at this stage, performed laser capture microdissection of the lobular epithelium to try to understand which proteins were affected after this anti-progestin treatment. And we did this by sectioning FFP blocks onto membrane slides, and then we used laser technology to capture the lobular structures to perform protein purification and perform the data analysis. And by doing this, we identified 65 differentially expressed proteins after treatment, and the pathway analysis using Reactome identified extracellular matrix organization, as you can see, as the most significantly altered by anti-progestin treatment. And then we looked at the extracellular matrix proteins. We found that 27 out of these 65 differentially expressed proteins were actually extracellular matrix proteins, and they were all downregulated. Many of these proteins were collagens, as you can see, and interestingly, if you can point, collagen VI proteins were among the most significantly downregulated proteins after treatment. So based on this data, we then decided to investigate collagen VI expression in relation to the localization of the luminal progenitor cells, here marked by SOX9. And we could actually find that collagen VI was found in close proximity with the SOX9 positive cells, here represented in red. And we were then able to quantify this by single cell neighborhood analysis, in which we identified the SOX9 positive cells, here represented in red, and compared them to the SOX9 low cells represented in yellow. And we could confirm that there was increased collagen VI expression in close proximity to the SOX9 high cells. So since then, collagen VI has been shown to play a role in fibrillar collagen organization, we have then decided to use picroseries red staining and polarized light to quantify the alignment of collagen, and we found that there was a reduction of collagen organization. This very consistent after this anti-progestin treatment. And here I've exemplified this on these boxes showing the in the polarized light. You can see the elliptic shape reflects high collagen organization, and the circle shape after the treatment with this anti-progestin drug reflects low collagen organization. We know that collagen organization has been associated with tissue stiffness, so we selected 4 paired samples that had at least 10% reduction of collagen organization, and we have shown that there was a significant reduction in tissue stiffness using atomic force microscopy in 3 out of 4 of these paired samples. So at this stage, our question was, so does a stiff microenvironment can contribute to the expansion of these luminal progenitor cells? And this is where we have been very lucky to use the Well Biosciences collagen mimetic hydrogels that allow us to mimic what happens in the breast tissue, either at soft or stiff conditions. So we mimicked what happens in the breast tissue using these hydrogels, and we cultured breast organoids and microstructures in soft hydrogels and also in stiff hydrogels. So this is ranging from 600 to 900 pascals to 1800 to 3000 pascals in the stiff hydrogels. And when we did that, so we looked at the RAMP ligand, which is a gene that is downstream of progesterone receptor, and we found that there was an upregulation of this gene when we grow these breast structures in the stiff hydrogels. And we also looked at the luminal progenitor markers, SOX9 and c-KIT, and we found that there was a clear increase of the expression of these luminal progenitor markers when these organoids were cultured in the stiff matrices. So we then also wanted to culture again these breast organoids in stiff hydrogels, and we have shown that when we did that they have increased expression of luminal progenitor markers c-KIT and SOX9. But when we then treated these organoids in stiff conditions with an anti-progestin drug, we could see that we were able to block the increase of the expression of these luminal progenitor markers c-KIT and SOX9. And we could also observe, when we look at mammal cell formation efficiency, that, as I said before, it's a functional assay to measure luminal progenitor activity, that when we've grown the organoids in stiff conditions and then dissociate the organoids and plate single cells, we could see that those cells that were grown at stiffer conditions were able to form more of these colonies that we call mammospheres. So saying that, meaning that we have increased luminal progenitor activity. But when we treated those organoids in stiff conditions, but in the presence of an anti-progestin drug, we could again block the tissue stiffness induced luminal progenitor activity. So then we went back to our own study and we looked at the RNAseq of our baseline samples, and we then separated our samples in high or low mammographic density. So AB corresponds to low mammographic density and CD corresponds to high mammographic density. And when we did that, we observed that the women that have higher mammographic density at baseline have increased expression of 5 genes, and 2 of them, as you can see there is the RUNC ligand and chemokine ligand 13. And these are genes that are secreted by progesterone receptor positive luminal hormone sensing or luminal mature cells. So meaning that we have activation of progesterone receptor on those women that have high mammographic density. So we then went back to the results with the anti-progestin treatment, and indeed we found that our results were more relevant in those women that have higher breast density compared to those women that have low breast density, as you can see here, looking at in terms of proliferation and Ki67. So this means that, and this indicates that breast density might be a potential biomarker of response to anti-progestin drugs. So as a summary, what I have shown you was that the breast tissue of these women that are at increased risk of developing breast cancer, they have increased numbers of proliferating luminal progenitor cells, here marked in orange, the SOX9 positive cells. And these cells, marked in orange, they are in close proximity with collagen VI, represented in red, and these fibrils more dense around next to the orange cells. And these cells, luminal progenitor cells, receive signals from those cells in the dark blue that are the luminal mature cells, but they also, these luminal mature cells also send signals to fibroblasts and basal cells. And so when we use an anti-progestin treatment, we are able to block those signals that are coming from those luminal mature cells represented in dark blue, and this leads to a collagen-driven extracellular matrix tissue remodeling, and eventually then leads to a decrease and a reduction of the luminal progenitor cells, their proliferation, a reduction of the collagen organization, and a reduction of the tissue stiffness. So in conclusion, next, what our data is showing is that anti-progestin drugs are a promising strategy for the prevention of the luminal progenitor-derived breast cancer. And as I said in the beginning, we were able, after almost 10 years of work, were able to publish this work end of last year in Nature. And I just would like to finalize by thanking first, if you can move to the next slide, please. First of all, I would like to thank all the women who have very altruistically volunteered to participate in this study and who have donated all the breast tissues that we have used in our studies. Of course, I would like to thank all my colleagues and collaborators in Manchester, in particular Sasha Howell and Rob Clark. I would also like to thank all our collaborators around the world. In the UK, the team from Walid Khaled, in Italy, the team from Sebastian Ando, and in Toronto, in Canada, the team of Ramacoca that has been very useful for the analysis of the single-cell RNA-seq. Of course, this work could not be done without the funding bodies, so thanks for their support as well. And thank you all for listening, and apologies for the technical issues.
Thank you, Bruno, Dr. Bruno Simões, for your presentation, and it was an excellent presentation. And we do apologize for the technical difficulties, but now we will still go into our Q&A. We might run a little bit over, but it's okay because of our difficulty we had. We had a question here. So why are, Dr. Simões, why are luminal progenitors, LP cells, considered a key target for breast cancer prevention?
So yes, these are the cells that we know that are proliferating, and that are proliferating due to the signals that they are receiving from these luminal mature cells. And these cells have also been shown, the ones, recently, the ones that are more likely to develop and accumulate mutations because of their higher proliferation rates in response to signals. So they are the cells that are most likely to accumulate mutations, and hence have the ones that would eventually lead to breast cancers. And this has been shown, for example, looking at the most aggressive tumors that are the triple negative tumors, have been shown that the signature, the gene expression signature of the luminal progenitor cells is similar to the signature of these more aggressive tumors. So it's believed that those more aggressive tumors have the origin on these luminal progenitor cells, hence the target we would like to not have those cells, or have as low number of those cells as possible in those women that are at increased risk of breast cancer, so that they are not able to form tumors.
Thank you. How did anti-progestin treatment affect the breast tissue microenvironment, or stroma?
So as I was pointing out on the summary slide, we observed, and it's more clear in the study, and I didn't have time to go through all the data today, but clearly the ligands that are coming from the progesterone receptor positive cells, that are the luminal mature cells, are not only impacting in the epithelium, but they are also impacting on the stromal cells. So for example, fibroblasts and myoepithelial cells. For example, we found a very important ligand, Wnt5a, that is clearly by the single-cell RNA-seq analysis, seems to be clearly affecting the fibroblasts and the collagens, particularly collagen VI expression. So there are ligands that are coming from the luminal mature that, in the presence of this anti-progestin drug, are blocked or inhibited, and therefore are not affecting and not producing those normal effects that they would be producing in those stromal cells. So that's how this drug is affecting the stromal as well, by inhibiting progesterone receptor signaling and the downstream target genes that would be affecting the stromal cells.
Thank you for that. I think this is probably related to the same thing, but what is the purpose? What is the proposed mechanism for how anti-progestins reshape the breast ECM?
Yes, exactly. So that's exactly that. So that's what we observe is that the factors and the ligands that would be coming from these cells that are progesterone receptor positives are having a major impact on the other cells. We didn't speak, for example, today in the immune cells, but maybe also in the immune cell compartment there will be effects. But our data shows, as for now, is the effects that are very clear both in the fibroblasts, and that's how they are reshaping the stroma by decreasing the collagen production on those fibroblasts. It's then able to reduce the stiffness and the collagen organization that is around those epithelial cells that would then lead to breast cancer. So we are reshaping the collagen organization and the stiffness around those the epithelium, and in particular those cells that would be the origin of breast cancer.
Oh, great, thank you. It's a great one here. What is the main future clinical implication of this study's findings?
So, of course, first of all, what we would like to do is to pursue these to bigger studies, and we would like to compare these anti-progestin drugs, for example, this one, ulipristal acetate, or or other anti-progestin drugs in comparison to current therapies that are already approved to use as a prevention therapy of women that are at increased risk. And for premenopausal women, the only drug that is approved is tamoxifen. So we'd like to do a study in which we compare side by side the anti-progestin drug with tamoxifen, and for a longer period, hopefully up to 1 year. And the primary endpoints of these types of studies would be, first of all, safety. But looking at our data, the other endpoint would be, of course, mammographic density, and seeing if there is an effect in the reduction of mammographic density with our anti-progestin drug in comparison to an anti-estrogen drug like tamoxifen.
Thank you. Okay. I have a few more questions, but I don't want to go pretty much over, but I'll see if I can combine these two questions together. With the VitroGel collagen system, I think you've already talked about how did modeling progesterone signal in vivo, and one is were you were able to change the ECM by adding growth factors or not. I'm not sure if you're, I'm trying to combine the two questions together, but and how long, I don't know which, if from our team or from you, Dr. Simões, how long can patient-derived organoids be cultured in the VitroGel system? I think that's probably for our team here.
Yes, I'm happy for you, but we have grown them for up to 14 days, but this study was done for 7 days. So 1 to 2 weeks, the organoids are still viable, and these experiments can be done. But as we have done here, in 1 week, cells are completely viable, and the organoids, and it's completely fine to use that. It could go up to 2 weeks in these studies, but we have not taken this further than 2 weeks. And just say, and you were mentioning as well how we have used this to mimic the stiffness. This was very useful, the hydrogels, because by using the hydrogels we can mimic what's happening in the breast tissue, and the stiffness that we are using, it matches what has been previously reported to match what's the stiffness that are found in women that are either at a low or high mammographic density. So with these hydrogels, we have been able to mimic that and then assess the effects. And some of the effects that were quite surprising was the activation of progesterone receptor on itself in the absence of progesterone ligands, just by the stiffness itself. So that's something that we would like to pursue as well.
Perfect. Yep. That is, I'm going to pursue that will be the last question for the end of our session here. Thank you, Dr. Bruno Simões, and thank you everyone for getting support of this webinar. We do apologize again for the technical hiccup, but as you know, tech is like that as well. For more information about Dr. Bruno Simões' research, there are the last slides here. You might see his name. Please reach out for his, great work and his questions about the published paper in Nature as well, and connect with us as well in the World of Bioscience on our LinkedIn page to find more about our hydrogels, VitroGel cell culture applications, our Xeno-free end-to-end workflow, and how we can help you as well as we help Bruno and his team, not just one of his team. How VitroGel can be used in real-world applications for clinical studies, with a Xeno-free material that can be used. Thank you so much again. Thank you, everyone. Dr. Simões, thank you again for your work and your presentation. Everyone have a great day. Thank you so much.
Thank you, Christopher, and thank you everyone.